So I'm going to be talking to you about a transdisciplinary consideration of sandflies and Leishmaniasis. But before I start, I'll just give you a warning. There will be some gunfire during my presentation. Okay, so I'm going to be talking about some of my research over the past years, but also about some of my sort of public engagement activities. But first of all, I'm going to focus on my research that I've done. I'm basically an insect microbiologist. I've worked on insects as models to study the role of gut microbiota and their host interactions. And today I'm going to be focusing on my work on sandflies as vectors of the medically important Leishmania parasites. The sort of the third final part of my presentation, I'm going to be talking about some creative projects that I'm involved with, working with different artists on microbes and insects. I'm quite interested in how, as scientists, we should be influencing the public debate within society, and the fact that we have a responsibility to contribute to society, just not in our science, but also in cultural aspects of life throughout the country and throughout our world. So when I was a student, way back in the dim mists of time, came across this textbook about insect microbiology published in the 1960s, and a well-known American microbiologist gave this quote: "A comprehensive understanding of the biology of insects requires that they be studied in an ecological context with microorganisms as an important component of the system." And that struck me at the time as a student. Yeah, that makes so much sense. You know, we shouldn't just be looking at insects on their own as an entomology discipline, or studying microbiology on its own. We should be bringing the disciplines together. And I've sort of spent my career gradually moving across the disciplines until I'm sort of encompassing as many as possible. So I started my work as an insect microbiologist studying locusts. I developed a germ-free rearing system with locusts to look at the protective effect of gut microbes and insect pathogens. We developed a Gnotobiotic isolator system. This was in the 80s, and we were able to rear these locusts through many generations. They were quite healthy, with a sort of vitamin supplement in their freeze-dried grass and bran, and we were able to inoculate these locusts with individual bacterial strains to look at changes in the microbiology, the behavior of the locusts, and in the volatile components produced by these insects. We came up with some interesting information, which I haven't got time to go into in this talk today. We're welcome to discuss that. So then I moved to working on medically important insects, sandflies, and the thing that immediately struck me about sandflies, from my experience as an insect microbiologist, was that, well, these sandflies actually possess a unique set of features that are interesting from a microbiological perspective. Because, of course, Leishmania live inside the gut of the blood-sucking female. They don't have a sort of phase where they go through the hemolymph to the salivary glands, for example. They develop in large numbers inside the midgut of the sandfly, and then they move up to the foregut, and then they're ejected during the next blood feed of the female fly. So they're a component of the gut microbiota, essentially, of the sandfly. Sandflies are also blood feeders, the same as similar to mosquitoes. So they're able to acquire different microorganisms from plants as well as from animals. It's only the female that feeds on blood for egg production. And finally, soil-dwelling larvae actually feed on a sort of microbe-rich compost, like annual burrows or the bottom of tree buttresses, etc. So they're able to acquire a whole diverse range of microorganisms, some of which are potentially transmitted through the pupal stage to the adult. So the Leishmanian population lives in the sandfly gut, in the vector, and if you label some parasites with a fluorescent tag, you can actually visualize, if you put your infected sandfly under UV light, you can actually see the large Leishmania population in the thoracic midgut of this fly. So what is Leishmaniasis? So you have Leishmaniasis in cases in the south of France and Mediterranean. It's a so-called neglected tropical disease, so funding's quite difficult. I don't make life easy for myself in my career, so, but we managed to get funding. But the disease itself is in three main forms. It's visceral, cutaneous, and mucocutaneous leishmaniasis. There are up to 1 million or so new cases a year found worldwide from Central South America, Eastern Africa, Southern Europe, Mediterranean, the Middle East, of course, and Asia. So it's very widespread, around 30,000 or so deaths a year through mainly visceral leishmaniasis, but that's probably an underreported figure, with very high amounts of morbidity and life lost through suffering in terms of also mental health and stress, for example, through cutaneous leishmaniasis, which can be quite disfiguring, and we're seeing that quite a lot in the Middle East, for example, in Syria and Afghanistan. So around 20 or so Leishmania species involved, and as I say, they're transmitted by phlebotomine sandflies, although we're working on some evidence that there may be also some midge species involved in transmission of Leishmania. But there are also a number of natural animal reservoirs as well, around 70 or so species, of which the most significant are dogs. So sandflies, well, phlebotomine sandflies, a tiny insect. This is a colleague's thumb, and so you can see sandflies are pretty small. They're smaller than Drosophila. They've got this characteristic V-shaped wings that you can see. If you see a sandfly sitting on the wall, you know, oh, that's a sandfly. And they're quite silent in flight. They're light brown, so they're difficult to see. I had the privilege of having some in my house in Tuscany when I went on holiday last year, so that was a treat. And there are about 800 species or so of these sandflies, of which around 78 transmit Leishmania. So as with many of these diseases, the vector-borne diseases, we very much need an interdisciplinary approach to reduction in cases of the disease, and that is truer than ever with Leishmaniasis. And so, of course, we have groups working on drug development, vaccine development, but I'm particularly interested in working in the area of disruption of transmission via the vector, but I'm also very interested in starting to work more in community participation, co-design with local communities, community education, because at the end of the day, without the involvement of the local population, it is very difficult, as we see, for example, with the dengue work. It's very difficult to get significant success. You can have very good technologies, very good ideas, but they may not fall work on the ground, depending on, for example, cultural practices. So sandfly research has been going on since the early 1900s. the discovery of sandflies as vectors for Leishmania. Up until the 1970s, 80s, really, there was this sort of bipartite analysis of the Leishmania parasite and how it developed inside the sandfly. So when, really in the 1980s, still carried on, I first started working on sandflies in the 90s, and sort of I came across a sandfly expert, and he said to me, "Why are you bothering to work on gut microorganisms, bacteria and yeast in sandflies? The blood meal is sterile in sandflies." So it's great puzzlement from people. At the time, there was this sort of quite a division between entomologists working on their discipline and microbiologists working on theirs, and sort of not a lot of overlap between these groups, but happy to see the things have changed remarkably in the last 10, 15 years. So in the 90s, we did our first sandfly microbiota studies, and we started to work on bacterial interaction with Leishmania. Now in the last 10 years, we've seen quite an increase in interest on sandfly microbiota studies, which I'm going to be talking about in this presentation. So I don't expect you to sort of see much of this in detail. I've just put this up really to give you an illustration that we now know a reasonable amount about the gut microbiota of sandflies. So this is a network analysis done by Eric Telleria and others based in Fiocruz in Rio. They collected data from lots of publications, lots of work being done on, in this case, Lutzomyia longipalpis, the South American vector for visceral leishmaniasis. And so, for example, you can see bacterial genera which are located both in blood-fed insects and in infected insects. So the message here is we are starting to understand quite a bit about the gut microbiota of sandflies, but there is still more work to be done. So our model system that we use at Lancaster is the South American vector for visceral leishmaniasis, Lutzomyia longipalpis, vector for Leishmania infantum. Interestingly, Leishmania infantum was introduced to South America via probably dogs brought over with the Spanish or the Portuguese to South American continent. So there's another European export. And this is visceral leishmaniasis in South America. Lutzomyia longipalpis is a very good insect to work with. It's the sort of Drosophila melanogaster of the sandfly world, if you like. So it's relatively easy to work with, and it's interesting because it acts as a permissive vector. It allows the development of different Leishmania strains within its gut. So we can do gut microbe experiments. We've established a membrane feeding system for maintenance of this sandfly colony, which is most sandfly colonies have to be reared on using animals, anesthetized animals. We've now been using this membrane feeding system for the past 10 to 15 years. So we can use that for maintenance. We can also use it to do infection experiments. So here we've got 4 cages with the Hematec membrane feeding system, but we have to use chicken skin rather than parafilm as they do with mosquitoes, because they won't feed through parafilm. They need some sort of live skin in order to get interested. So we can inoculate this system. We can add bacteria or Leishmania, for example, to study the infection process. So we asked the question, does the gut microbiota influence Leishmania development in sandflies? Here, I've given an example of some experiments that we did, looking at what we call colonisation resistance, thinking about the gut as a sort of ecological niche, if you like. And we looked at the effect of yeast and bacteria, which were first isolated from wild-caught sandflies in northeast Brazil. We brought them back and inoculated them into newly emerged sandflies, and then we challenged those insects with Leishmania species. And to the left here, you can see a number of parasites per fly with different bacteria or fungi which have been inoculated into the sandflies. And in some cases, you can see a significant reduction in the number of promastigote parasites per insect, depending on the microorganism already occupying the insect. And this translates into a reduction in number of Leishmania infected sandflies, depending on the presence of different bacteria or yeast. So also, there was the question of, can also some gut microbiota potentially benefit Leishmania development? And so this work was taken up by the sandfly vector group in NIH, led by Sheldon Kamhawi and Jesus Valenzuela, with Kelly and others, and they were actually having some difficulty in infecting their sandflies with Leishmania, and so they also became interested in the gut microbiota. They found also, as we have found, that bacterial diversity progressively decreases during Leishmania infantum infection population growth in the gut of the sandfly. But interestingly, certain bacterial species actually became more dominant as the Leishmania population increased. And also it seems that certain bacterial genera may actually be important in benefiting and helping the development of the mammalian infective form of the Leishmania parasite in the gut of the sandfly. There's still a lot of work to be done on that, but it's really intriguing potentially that there can be costs and benefits to the Leishmania population depending on the different microorganisms present. So I think the idea is that the microbial composition within the sandfly is very potentially important and has some control potential. So what I'm saying here is that the species that initially occupy the sandfly as it newly emerges as the adult is potentially a critical determinant for successful Leishmania transmission. So we started also to think about the costs and benefits of Leishmania to the sandfly. Can Leishmania be beneficial to the sandfly host? Indeed, we asked the question, are there circumstances in which Leishmania can protect the host? Because there could be some dual benefits there. So we set up a series of experiments to look at whether Leishmania will protect the sandfly against a bacterial pathogen. We infected sandflies with Leishmania, established the population in the gut of those insects, and then we challenged those sandflies daily with an insect pathogen, Serratia marcescens, which is reasonably pathogenic towards the sandfly, and then we recorded sandfly survival. So on your left here, you see some sandfly survival experiments days after challenge with bacteria. And so I'll highlight two lines. The pink line here is the population which has been just blood-fed, but challenged with Serratia, and it's a drastic reduction in the population of those sandflies after 6 days. In comparison, where you have sandflies which contain a Leishmania population. As you can see here in the black line, they survive in the same extent to the controls. So this is pretty impressive. In fact, also if you do, for example, you can work in vitro with Leishmania, and if you grow them together with Serratia, the Serratia will kill the Leishmania. And so we see improved sandfly survival, and also intriguingly, we see the same numbers of Leishmania within the gut of the Serratia challenged vector as we do for the ones that were not challenged with the bacterial pathogen. So really, it seems that under some potential circumstances, Leishmania could potentially benefit sandflies. And this is quite interesting. You can sort of speculate on biological control and how that could potentially have sort of adverse effects. It could actually promote the survival of the sandflies containing the Leishmania. So it's something to think about in terms of overall microbial ecology. So we were also starting to think, well, I guess when the Leishmania are sent into the mammal, perhaps some bacteria are also co-located, and NIH group again did some fantastic work. They looked at gut microbes transferred by Leishmania-infected sandflies, and they found that they actually prime the immune system of the host. So here's a schematic produced by Day et al. And for example, you see here that gut bacteria which are co-injected with Leishmania into the hamster actually trigger inflammasome, and this amplifies interleukin, which sustains recruitment of neutrophils to the bite site. In contrast, when you give antibacterial compounds to sandflies with the Leishmania, then sandfly, during the transmission process, there is an abolition of neutrophil infiltration, and that leads to reduced spread of Leishmania through the mammalian body. So now we've got to the stage here where we can see that gut microorganisms allied to the presence of Leishmania have a potential effect on the not only the development of Leishmania, but also the transmission process and the actual disease outcome within the mammalian host. So this is where we are now. I think we've shown the central importance of studying microbial ecology of the vector, and there's this important interplay between positive and negative effects on the fly, the Leishmania, and indeed on the mammalian host. So we have these other microbial passengers with Leishmania which are determining success of transmission. We know that Leishmania potentially can reduce sandfly longevity during certain periods of stress, but potentially they can also benefit the fly as well. So that's my work on the science side of things. I'll talk a little bit now about some creative projects I've been involved with. So hopefully at the moment, up till now, I've sort of engaged with you with a clear factual presentation. So from now on, I'm going to attempt to engage you from sort of artistic perspective, so less directly through trying to engage with the emotions. So I'm going to be talking about a few projects here, something called Tropical Tales, a children's audio story, something called the Lambanana, which is a sculpture created in Liverpool as an iconic comment on genetic modification. It's a lamb fused with a banana, and I had to sort of use that sculpture to display the life cycle of Leishmaniasis, and a bed nets, not bombs, bedding art performance that I did in the style of John Lennon and Yoko Ono, a comment about how insect vectors take advantage of the human condition to actually kill themselves. So when we go to warfare with our species, insect vector species love to take advantage of the changing environment. And so it's also a comment about the arms industry versus funding for tropical medicine. And finally, digital artwork that I'm doing at the moment called Parasite Seeing, life from the perspective of both sandflies and Leishmanian parasites. So this is an audio story which I developed with a group of artists. Hopefully it will work. Okay, so this is a sort of plastic piece, and you can use an audio pen to actually touch on different components of the story. And with an audio pen, you can actually have this story in 5 different languages. So basically, the story is of an Indian brother and sister, Raju and Anju. Anju gets visceral leishmaniasis, and Raju goes off to search for a cure for leishmaniasis. He falls asleep, he dreams he's eaten by a sandfly, and he's actually at one stage being attacked and fighting with a Leishmania parasite, and then the sandfly eats a plant, and the plant material kills the Leishmania parasite. Raju wakes up and he finds the plant and he creates a cure for his sister, and they live happily ever after. So this is a Lambanana sculpture. You've seen these sculpture trails, for example, in Berlin with cows, and I'm sure in Paris as well. And so my PhD student and I volunteered to create a sculpture with Leishmania drawn on the side here. This is a community participation project, and at the front you can see a naked lady who is acquiring Leishmaniasis, and here we have a student. We had 180 people actually participating, putting a finger dot around each parasite as a part of a sort of community art project. And here we see the essential elements of a deconstructed sandfly. And here there's the characteristic wing veination of Lutzomyia longipalpis. Then we had bed nets, not bombs protest. And I spent a day in bed in an art gallery in Liverpool, and warning, there is gunfire. So there's 15 more minutes of that. I don't think we can carry on. So finally, a digital artwork that you can access called Parasite Seeing. It's thinking about how Leishmania and sandflies were happily going around their business for millions of years until the arrogant human pulled them out and put them into the lab and studied them over the last 100 or so years. So there's different social media things you can look sort of life from the perspective of a parasite in my lab. Also Leishmania, it's a sort of confocal travelling through, sandflies searching for egg-laying sites in the Tuscan countryside. And this is LD Bob, my alter ego. It's a Leishmania parasite on Twitter. Thank you.
One quick question. Otherwise, we'll continue. So you have to jump up right now, or yes.
Very nice, Rod. I have a question, just a quick question about when you did put together bacterium, like you had this, only one, and then you mixed two, and there's the Ochrobacterium you did not put together. There was a synergy with that too. It was a reduction if you put together or not?
Yeah, we didn't try it with Ochrobacterium, but I think potentially there will be a synergy when you add two or more species together. So that sort of makes sense to me. So there's a lot of work to be done. Very few sandfly labs in the world. There should be one in Institut Pasteur, of course. And I know some scientists there want to have that. So.
Okay. After this message, I think we continue. Okay. Thank you very much.
Okay, thank you.
The next and last speaker of this fifth session is Philippe Bastien from the Institut Pasteur, and he'll talk about the trypanosome journey in the tsetse fly.